Hello everyone, myself Dr. Ashwath Narayan R, lecturer in genetics. In today's class, we're going to discuss about disease risk prediction using Hardy-Weinberg equilibrium. Before going to the disease risk prediction, we're going to discuss about what is Hardy-Weinberg equilibrium. Hardy-Weinberg equilibrium is a imaginary formula which is proposed by two eminent scientists called as Hardy and Weinberg. So, this principle of Hardy-Weinberg equilibrium states that genetic variation in the population will remain constant from one generation to other generation in absence of these disturbing factors. We're going to discuss about the disturbing factors further. In a population, allele and genotypic frequency are constant. We're going to discuss about allele also. What is allele means? It's an alternative form of genes which is present in homologous chromosome, which mainly code for some phenotypic character. This is called as allele. This is called as allele. That means if you see the image here, some coding part is here and the other coding part is there. Whole these two are homologous pair, and which these two coding genes which code for something, some phenotypic trait. That is called as allele. Allele is alternative form of gene that is present on homologous chromosome, which code for one phenotypic trait. So these allele as well as genotypic frequencies will be constant unless there until there will be some disturbing factors or evolutionary forces acting upon them. You want to know about Hardy further? This scientist who is the main person, who is the main person who mainly encouraged Srinivasa Ramanujan, Indian mathematician, to publish his thesis in reputed institution. He's the first, he's the main person who is mainly contributed for Srinivasa Ramanujan to publish his thesis. Further, some conditions are there. First, we need to know about this whatever formula is there, which is put forward by Hardy and Weinberg scientists, is equal to p square to p and plus 2pq plus q square is equal to 1. We're going to derive the equation further. How they got this equation, and some conditions are there for this formula to be valuable. For that, we need to know about what are the assumptions they made to propose this theory. These are the 7 assumptions they made to propose the theory. That means this Hardy-Weinberg equilibrium only applicable for diploid organisms. That means whatever mammals, human beings, other reptiles, they mainly participate in sexual reproduction. So the animals which participate in sexual reproduction, animal as well as plants which participate in sexual reproduction, so they are applicable under Hardy-Weinberg equilibrium. And second point is same. That means the organism should be participate in sexual reproduction to apply this Hardy-Weinberg equilibrium. And the generation should not be overlapped, and mating should be random. So mating should be random means in same population mating should not be happen. So that's a third and fourth conditions. And the population size should be larger, extremely larger population should be there to apply this formula. And as I said earlier, the gene and allelic frequency should be constant. And last one is, these are whatever we discuss about some factors which mainly influence on whatever gene frequency and allelic frequencies are there: gene flow, migration, admixture, immigration, selection, mutations. These factors will not influence on this equation. These are the 7 assumptions and conditions will be there to apply Hardy-Weinberg equilibrium. So Hardy-Weinberg equilibrium, by assuming these factors, they put forward this formula here. P plus q is equal to 1 and p square plus 2pq and q square is equal to 1. These are the two formulas they put forward it and derive the equation with respect to the population, where they can calculate mathematically the allelic frequency, genotype frequency, gene frequency by using this formula. Whatever the value you will get by using this formula is somewhat imaginary. Why? Because this fact, this whatever formula is only applicable whenever the external forces like migration, immigration, mutations will not act upon the population. We're gonna derive the formula of Hardy-Weinberg equilibrium. So here, consider n is the total number of population. Then it should contains dominant allele, heterozygous dominant allele, and recessive allele. So total dominant, heterozygous, and recessive population will gives total number of the population in a particular geographical area. So why this is important means you can see here. So to calculate gene frequency in a population, genotypic frequency, this formula is very important, and to derive Hardy-Weinberg equilibrium, this point of the formula is crucial here. So always, we mention the number is 2n. Why? Because I already know that in assumption, the organisms which is participates in sexual reproduction, they are always diploid in nature. So because of that, 2n will be mentioned here. And D is dominant allele, H is heterozygous dominant, and R is recessive population here. Next, we move on to calculate the gene frequency here. So in gene, to calculate the gene frequency means genes. That means it may be dominant. Consider this is P and this is Q allele here. In that it becomes gametes here. This is gamete, and then it forms new gametes, and a new population will arise. So in that, this population will be, the population of this individual will be calculated by using formula is equal to a dominant allele is equal to 2 dominant divided by heterozygous divided by 2n. 2 dominant plus and heterozygous divided by 2n. Why 2D is mentioned here? You already know that in homozygous, this is the population. PP is there, P small p is there, and small p is there. In that you can see 2 dominant will be there. In that 2 alleles will be there. For that 2 dominant, 2D is there. Heterozygous, H is there. Only one H is mentioned. Why 2H is not mentioned means you can see in heterozygous population dominant allele is only 1 divided by total population. N is total population. 2n, that means diploid. Because of that 2n is mentioned, that is with respect to dominant allele. Next we'll move on to recessive gene frequency, and then you can follow the formula 2r is plus h is equal to plus h divided by 2n. This is recessive allele population. As same here, 2 recessive alleles will be there in recessive population, and then in heterozygous dominant, 1 recessive allele will be there, and 2n is diploid. This is with respect to gene frequency. Next we'll move on to genotype frequency. So you can calculate genotype frequency by using this here. Dominant allele, number of dominant allele divided by total number of the population. This is with respect to PP. That means homozygous dominant. Next one is heterozygous gene frequency is equal to heterozygous divided by a total number of population. Then recessive population, homozygous recessive population is there. Recessive divided by total number of population. Then how you derive the formula? So to derive the formula, you need to sum up P plus Q here. P plus Q is equal to whatever the gene frequency is there. Sum up this both here. 2D, 2R. Sum up that, and you'll get 2D and 2D plus H plus 2N plus 2R plus H plus 2N. So if you sum up that, so the common denominator is there. So common denominator available, and then if you sum up that, 2D, 2H, here H is there, H is there. If you sum up that, 2H will be there and 2R. So D plus H plus R is there. You already know that where you got this formula, D plus H plus R is equal to N. So here d plus h plus r is equal to n. N you need to mention 2n divided by 2n. So both are cancelled. Then 1. So p plus q is equal to 1. Then how's our p square plus 2pq plus q square should be 1. So I know according to Hardy-Weinberg equilibrium, consider p population, homozygous uh, population, one more homozygous population. This is gamete here. During mating they will become gametes here and become pq. If that is self cross, pq plus pq self cross with pq, then the population will be mentioned in Punnett square like this here. P here, Q here. If you cross that, so you will get PP here, PQ here, PP. Instead of P, you can see P into P, P square. Write P square here. Instead of PQ here, one PQ is there, two PQ is there. Write two PQ here. And then here instead of QQ, you can see Q square is mentioned. Understand? Then simplify the formula. Here p square plus 2pq plus q square is equal to p plus q whole square. You already know that, but from the previous value, p plus q in standard condition is 1. 1 to the power of 1, 1. So p square plus 2pq plus q square is also 1. So like that, so you can derive the formula for Hardy-Weinberg equilibrium. So, sir, it is for two alleles. What about for multiple alleles, sir? So, for multiple alleles, you can see p plus q plus r whole square is there. You already know the formula. P square plus q square plus r square plus 2pq plus 2qr plus 2rp. That is equal to 1. Further, we'll directly go with the problems here. The problems on disease risk prediction. First, the first problem is in the incidence of Marfan syndrome in a small village of US, the following number of genotypes are produced, mentioned below. So MM is given, capital M is given 152, capital M small m is given 53, small m is given 30. So total population is equal to 235. Then calculate gene and genotype frequency. First we'll go with the gene frequency. So gene frequency is equal to gene frequency will it is for P here. P is equal to 2D plus H plus 2N. So 2D 2 into dominant allele 152 plus heterozygous 53 divided by 2 into 235. If you simplify the formula, then you'll get 357. 357 you'll get it divided by 470. So 470 you'll get it, and if you simplify, you'll get the value of 0.75. Next, you'll go for calculating Q here, gene frequency 2R plus H divided by 2N. Then 2 recessive means 30 divided by heterozygous is 53 divided by 2 into 235. Then you'll get around value. That means if you calculate the value is nearby to 0.24. That is equal to 0.25. Then if the value is correct or not, you can sum up the p plus q here. That should be near to 1. 0.75 plus 0.25. So that is equal to 1. So then our value calculation is correct. Next we'll go to genotype frequency. Genotype frequency, then we'll go for p square here. P square is equal to dominant number of individuals who are dominant divided by homozygous dominant divided by total number of population 235. So 152 divided by 235, 0.64 is the answer. Next one, 2pq. 2pq is equal to 53 divided by 235. 53 divided by 235, the value will be 0.22. Next, q square is there. Q square is equal to 30 divided by 235. 30 divided by 235, the value will be 0.127. If you sum up these values, you'll get nearly one value. So we calculated gene frequency and genotype frequency here. Understand? So in same population, further these questions will be further asked like this. That means as a genetic counselor, suggest suggest this population with respect to the incidence of the disease. That means you already know that Marfan syndrome. Marfan syndrome is mainly inherited by autosomal dominant fashion. So in that population you can see 0.75, that means nearly out of 175 people is affected with Marfan syndrome. Understand? If you go deep here, 64% or 64% is already affected with Marfan syndrome. They are homozygous dominant. In that, 0.22 will be the heterozygous population. They are also included here. Understand? So total population is affected. Why? Because the Marfan syndrome is homozygous. Sorry, that means autosomal dominant inheritance. Only one allele is enough to cause the disease here. It may be PP or capital P, small p. Both will affect the individual. Understand? So because of that, so here whatever as a genetician for the population suggestion is who and all homozygous dominant will be there. They should not marry. They should not marry. Why? Because all of them will be affected. If they married to unaffected individual, take example here, PP married to small p or small q, assume that this is unaffected, healthy population. Then if you cross this, definitely all of them become PQ only. So here one allele is enough to cause disease. Because of that, if the affected individual is married to unaffected in Marfan syndrome, definitely all of them will affected with Marfan syndrome. Marfan syndrome is very much life threatening, and some complications are there. In some symptoms, like take example like cleft lip and palate. In that condition, only lip and then cleft palate will be affected. So they can marry to anyone, and then surgically they can be corrected. But in the incidence of Marfan syndrome, some severe complications will be there. So because of that, who and all affected cannot marry, should not marry to unaffected individual. uh, because of that, if they, you need to check the karyotype, and further you can proceed. If one allele is affected, two allele is affected. In case of one allele affection, if one allele is affected, so then if unaffected person is married, definitely QQ is there. Then I can observe that so half of the population is unaffected. There will be, you can check the karyotype, and then after mating, the fetus amniocentesis or chronic villus sampling can be done, and then we can check the incidence of the disease here. So because of that, the affected individual should not marry in further proceeding of the disease to the next generation. Next, we move on to second question here. The following genotype were observed in the incidence of phenylketonuria. Calculate the observed genotypic allelic frequency for this population, and suggest the solution for this population as a genetic counselor in prevention of inheritance of this disease here. That means observed genotypic frequency is asked here in this question here. The observed genotypic frequency is like this. We can calculate that. First, we need to sum up this PP 40, P small p is 45. Then small p is given. Small p is given. That is 50. So you need to sum up this, and you'll get the population of 135 total. Then we'll calculate the observed which is given here, expected population is different. If they asked calculate following genotypes were observed genotypic allelic frequency is given, then you can calculate directly here. That means just go to p square. So then 40, that means dominant divided by total population 135, you will get the value, that's 0.30. Then 2pq, calculate 2pq here. P is given, 2pq is equal to heterozygous population divided by total number of population 45 divided by 135. So you will get the value of 0.33. Then q square calculate. Then q square is equal to recessive population divided by total population 50 divided by 135. So you'll get 0.37. Sorry, 0.37 as an answer. 0.37. So that this is called as genotypic frequency. Next we'll go for gene frequency. Gene frequency, then this is observed genotypic frequency. Then we can calculate P is equal to 2D plus H divided by 2N. 2 dominant means 2 into 40 into plus 45 divided by 2 into 135. So if you calculate that, you'll get 0.46 as an answer here, and Q is equal to 2R plus H divided by 2N. So 2R means 2 into 50 plus 45 divided by 2 into 135. You will get answer of 0.54. 0.5. If you sum up this both, though, you will get the one value that is equal to 1. This is the observed the genotypic and allelic frequency. This is observed. So if they asked expected, observed is different, expected genotypic frequency means you need to go for a different formula, but we'll go in next video we'll discuss about it. Next the next question is the suggest as a genetic counselor in prevention of this inheritance of the disease to next generation. So in that whatever suggestion is that what the disease is the phenylketonuria. Phenylketonuria is autosomal recessive, that is inherited by autosomal recessive fashion. So you need to know the points here. Affected, they will skip generations. Unaffected parents can have affected children's. Only autosomal recessive, genotype will inherit and cause the disease. And then equal ratio of males and females are affected in that population. So you need to remember that as a genetic counselor, you need to prevent the inbreeding. So you need to prevent inbreeding and should go for outbreeding, vigorous outbreeding. Outbreeding means as I said in previous videos, we discussed about inbreeding, outbreeding. In the incidence of these autosomal recessive diseases, in some inbreeding, such as in some Muslims will marry to cousin, sister, brother, and then in some tribal populations they will marry to the same cousin, brother, cousin, sister. So in that case, if any incidence of these autosomal recessive diseases are there, assume that if the person is heterozygous carrier of the disease, married to same population who is also heterozygous carrier, because without knowing the genotypes, if they married to the cousin brother, cousin sister, there is incidence of one affected individual with this population. Because of that, you need to take care, because one is unaffected and two will be heterozygous carrier of the disease, and one is affected individual which causes phenylketonuria. So phenylketonuria is a very dangerous disease that causes several complications. Because of that, you have to prevent the inbreeding here, and we need to go for outbreeding here. In some Hindu traditions, you can see Swagotra marriage. Swagotra means if they belong to same gotra, Swagotra marriage is prohibited. Why Swagotra marriage is prohibited? It is scientifically disproved that because of marrying to other gotra, definitely we can prevent the disease, and the genotype of the further generation will be very stronger. We can that further generation doesn't have this disease, and they doesn't become carrier of the disease. So Gotra marriage is prohibited in some Hindu traditions here. Same Gotra marriage is prohibited. Next we move on to one more question. Uh, one more question here. So in United States, the incidence of albinism is 0.001. Predict the frequency of the population in this population who are heterozygous carrier of the mutant allele. That means what is given here in US. The incidence of albinism, you already know that the inheritance will be autosomal recessive fashion, it will inherit. Then the incidence of that is 0.0001. What is given to you? Q square is given is equal to 0.0001. Then calculate Q here. Q is equal to square root of 0.0001. Q is equal to 0.01. So if you calculate Q, then apply formula here. P plus Q is equal to 1. P is equal to 1 minus Q. That is equal to 1 minus 0.01. That is equal to 0.99 is P value. P is equal to 0.99. Q is equal to 0.01. Then what is the question here? Frequency of people who are heterozygous carrier of the population. Heterozygous carrier means you need to calculate 2pq. Why? Because 2pq has both the allele, dominant allele, recessive allele. 2 into, we already know the p value 0.99 and we already know the value of q here 0.01. So the value of that population, heterozygous population is 0.10198. Okay, this is the question here. Next we'll move on to one more question here. About 90% of the population are tunkles. Tunkle is, the inheritance also given in some examination, in CSR examinations. In some questions they will give inheritance, right? Inheritance is given. So if inheritance is not given, definitely you need to move, you need to go with, you need to know the inheritance first. Otherwise, you'll miss out the question here. That means some few of the questions which inherited by autosomal dominant, X-linked dominant, autosomal recessive, X-linked recessive. You need to remember that if the inheritance is not given, in 60%, 70% they will give the inheritance. Otherwise, in some occasions, if they didn't give, definitely you need to remember some diseases. Here, tongue curl is given. Tongue curl is autosomal dominant inheritance. And what are the gene and genotype frequency for T and T here? So what is given here? 90% is tongue curl. That means 90% of the population. That means p square plus 2pq is given, or both is given here. 10% is unaffected with tongue curls here. That means they are non-tongue curls here. Non-tongue curl is given here. Not given, they are indirectly given. That means is equal to, if you convert that into digits here, 0.10 population is not affected with tongue curls here. That means q square is given 0.10. Then q is equal to square root of 0.10. That is equal to q is equal to 0.316 is tongue curls here. 0.316. Then calculate p here. Apply p plus q is equal to 1. P is equal to 1 minus q here. That is equal to 0.684 is the answer. Then you can calculate already gene frequency is given here. P is calculated, Q is calculated, that is gene frequency. Next we'll move on to genotype frequency. Genotype frequency is equal to P square. First we will calculate P square here. So P into P, we will calculate P into P here, 0.684 into 0.684. You'll get the value of P square 0.468 P square. Then 2PQ, calculate 2PQ. P, you know the value of PP 0.684, 0.316. So 2PQ value is 0.432. Next Q square value is given. So 0.316, 0.316. So you'll get the Q value, and Q square value is equal to 0.10. If your answer is correct, you just sum up these values, and that should be nearly equal to 1. Next problem is the last one. Last problem is the frequency of dominant allele is 0.6. Instead of dominant allele, if some disease is given, so apply some disease here. Frequency of, instead of dominant allele, just go for frequency of the Marfan syndrome or some other diseases with respect to autosomal dominant inheritance. So here dominant allele is 0.6, or frequency of Marfan syndrome allele is 0.6. What is given here? It is given here is frequency of dominant That means p square plus 2pq is given. P square plus 2pq is equal to 0.6. Right? P square plus 2pq is given. Sorry, pardon me. So p is given 0.6. It may be p square plus 2pq. Right? Then calculate the population. Population who are showing dominant character in 300. In a population of 300. So it may be homozygous dominant, heterozygous dominant. Because of that, we will directly calculate first Q here. Q is equal to 1 minus P, 1 minus 0.6, that is equal to 0.4. Q is 0.4. Then, so population of the dominant, recessive allele, we can calculate. Recessive allele population, population is equal to Q square into the n number population. Q square is equal to 0.4 into 4, 4 4s are 30, 1 6, sorry, 0 4 4s are 16. So if you calculate that, so Q square is equal to 0.6, 0.16 into n number of population 300. For the population you are calculating is 300. So here, if you calculate that, the number of individuals who are recessive characters is 0 and 48. Then you have to calculate the dominant allele here. The number of individuals who are showing dominant character is equal to total population minus the recessive population. 300 minus 48 is equal to 252 is the population who are showing dominant character. It may be p square plus 2pq. But because we don't know this population is homozygous dominant or heterozygous dominant, so this is the answer.